we're interested in the microbiome. So, right, so these bacteria that live on and in the body, but there's a big mystery there. So there's a lot of work that's gone into correlating differences in the types of microbes or what they do in a variety of disease states. But at the moment, we really lack a lot of technologies to be able to probe causality there. So to really figure out what types of microbes are impacting a certain disease, and maybe more importantly, what types of molecules they're making and what types of proteins that they're making that might affect those disease states. So the bulk of our research program is focused on microbiome engineering. So developing those types of technologies so that you can control the microbiome, so that you can add a given bacteria at will, take one away, add new genes or new functions to bacteria that are there. And those have applications in trying to understand how the microbiome works in a variety of disease states, but also has more future-looking applications too in trying to engineer bacteria in the microbiome that could sense disease states and produce therapeutics on demand as sort of these sentinel cells that might exist within your body that could dynamically sense and respond and cure different diseases. So there aren't too many mechanisms that are amenable to these types of high-risk, high-reward projects that I propose for the Beckman Young Investigator Award. And engineering the microbiome is challenging. There's not a lot of technologies to do so, and sometimes you need to take these big swings. So having sort of the ability to get the funding to put several students on these tasks of trying to deliver new gene cassettes within the microbiome was something that really pushed my research program forward in a way that probably wouldn't have been possible through other conventional funding mechanisms. So the Beckman Young Investigator Award funded Jay Fuerte Stone, who is a PhD student that recently defended their thesis in my group, throughout the entirety of their PhD, and also funded Josh Glazier, who is another PhD student who also recently defended for part of his PhD as well.
I think that the big impact is, of course, being able to fund really novel and cool research in the lab. I know that's one of the reasons that I joined was because this phage project was proposed and funded by this grant. So having this very cool project that already had money behind it, so I didn't have to worry of is joining this new lab with a professor that I would have been one of the first students. It was a lot less of a risk. And so I know that just having that kind of funding and then also having support behind a PI is a big deal, and it definitely made an impact.
The main thing that funding does is it provides us as scientists freedom to do the experiments and pursue the ideas. Mark in particular has had this idea since he started. And so when I joined, there was no real constraints on being able to explore this idea. My work in particular requires a lot of sequencing and a lot of submission of samples to get data back, and that takes resources to be able to do it. It's a lot of mouse work, and without solid funding, it's hard to plan long-term experiments. I think over my time here, I've done 30 to 40 different mouse experiments, and those don't happen without funding that allows us to ask interesting questions, maybe pursue something that we weren't necessarily positive was going to work, but was a little bit more exploratory. And over time, being able to repeat this and fail and still have the support to keep going leads to better science.
Arnold Beckman was obviously a pioneer chemist as well as an engineer in developing a number of techniques and instruments to be able to do measurements that no one had done before. And to be fully honest, going through a biological science lab, we'd see Beckman's name on centrifuges and spectrometers and stuff like that, but I didn't really have the full context of his contributions to science and engineering prior to having received the Beckman Young Investigator Award. So I was really surprised to see, these inventions that are spanning medicine and chemistry and biology that we don't even think of. We have pH meters in the lab, we have spectrophotometers in the lab, and sort of those core technologies that he developed have kind of existed longer than him and are kind of endemic throughout all biological research. But I also learned too, especially being part of this Young Investigator program, is sort of the ethos that he's carried with him as both a scientist and an engineer in trying to have a focus in sort of translating inventions that he made to make sure that they have a real world impact. And at least for myself, that's quite inspirational, thinking as an early career researcher, how I can sort of take my research that might be, a little bit further away from application, but always sort of keeping the eye on the prize in trying to make a real world impact instead of just making, fun things in the lab.